Olá, Esther, boa tarde.
Tá sem som, Esper.
Oi, Vivi.
Olá, professor, tudo bem?
Bem, a senhora tá bem?
Eu tô bem. Não precisa me chamar de senhora, não.
Vivi, tô brincando. Tá bem? Saudades da Vivi.
Hi, Melanie.
Did I say hi? Was I muted? Just wanted to say hi. Thanks for inviting me. Great to see you.
Hi, Melanie, welcome. Esper, your microphone is closed.
Apologize. Yeah, both of us were muted. So pleased to see you.
We should know by now, huh? After 3 years of doing this day and night, no? Hi, Jessica, nice to meet you. Really looking forward to your lecture.
Thank you. Nice to meet you too. It's a pleasure to be here.
Well, next time in person, guys.
Looking forward to it.
Hi, Professor.
We are with problem with our camera here, so I can't share our video with you, but you can see you. We can see you, okay? And you will start in 2 minutes.
So we carry on this retreat every semester. So this time, the idea is to go over several issues on HIV. So we thought that having you over would be a great asset to the meeting. So thanks for joining.
Really exciting. So it's a whole day meeting, or how is it? Tell us a little more. Is it and who is the audience?
This is our department, and because we live stream over YouTube, there are several people that join, but they do not interact in the meetings, but they can send messages over YouTube, so we can collect questions. But fundamentally is the Department of Infectious and Parasitic Diseases.
Fantastic. And so, Esper, your move is not affected by this. You're still part of that department, correct?
Oh, no. I'm part of the department, but I'm licensed temporarily because I need to take care of a vaccine manufacturing plant here in Sao Paulo.
I know. I was wondering how you juggle all this. So, well,
Angela, can you hear me? I was wondering who is going to check on the questions that come up from YouTube. Esper, do you know?
I think Angela and Valdir are taking care of those things at the hospital where we have a conference room. That's this lecture has been streamed to. There you go. At least I can see the front desk.
Angela, can you confirm if you guys can check the questions on YouTube?
Yeah,
I think
check the questions on the YouTube.
there's Angela. Yeah.
We will translate. Do you want the link to the YouTube, Vivi, to mediate the questions?
I can share I can share over WhatsApp to Vivi.
Well, I think that you can start now. Vivian will, that are a previous professor of our department, and now she is at United States and maintaining her collaboration with us. Thank you for being here to receive our guests. Please, we can start.
Hello, everyone. Thank you, Angela. Thank you, Dr. Valdir and the Department of Infectious Diseases for inviting me to be here today. It's my pleasure to be back at home and for a while, even from a distance. And I'm very pleased to introduce Dr. Jessica Castillo. I'll introduce Jessica first, since her talk comes first, and then I'll introduce Dr. Melanie Ott. So Dr. Jessica Castillo completed her medical training and her MPH at Johns Hopkins University. She is currently an assistant professor of medicine at the Division of Infectious Diseases at Vanderbilt University. And the long-term complications of chronic HIV infection, including immunologic consequences of aging in people living with HIV, are among Dr. Castillo's main clinical and research interests. She has developed very strong collaborations with leading Brazilian investigators, including us in Brazil, in São Paulo at USP. And she's now the lead investigator for ELEA Brazil, Estudo Longitudinal do Envelhecimento e AIDS, which she is going to talk to us about. Thank you so much, Jessica, for accepting this invitation. And I'd also like to introduce Dr. Melanie Ott. Dr. Melanie completed her medical training at the University of Frankfurt and her PhD at the Pickering Graduate School. She is now a senior investigator at the Gladstone Institute of Virology and Immunology and a professor at the Department of Medicine at University of California, San Francisco. So we are pretty close by now. Her research is focused on viral diseases, microbial pathogenesis, and immunology, trying to understand host human factors restricting or enabling these viral infections. And Dr. Ott is one of the lead investigators of HOPE, HIV obstruction by programmed epigenetics. This is a collaboratory study funded by the NIH, trying to understand and develop cure strategies for HIV. Thank you so much to both of you for coming, for accepting our invitation. I'll leave the floor now to Jessica Castillo. Thank you.
Thank you so much, Vivian. I'm going to share my screen. And someone just give me a heads up to make sure that a thumbs up, it worked okay. Did that show up all right?
Yes.
Okay, great. Well, good afternoon from Nashville. Thank you, Vivian, for that really kind introduction. It's really an honor to join you all today to talk about HIV and aging, improving both health span and lifespan in the Americas. I'd also really like to thank the conference organizers for the invitation to join you. I have no conflicts of interest to disclose. However, I do have a disclaimer. So in HIV research, the term older adult has generally been used to describe someone living with HIV who's over the age of 50 years. This isn't a term or a definition that I created, and actually may warrant some revision as the population living with HIV continues to age. But I do give this disclaimer when I'm speaking about older adults in this talk. It's generally talking about over the age of 50. Okay, so a little bit of a map of where I hope to take you all over the next 40 minutes or so. I really plan in this talk to take a hemispheric approach to discussing HIV, aging, and aging morbidity, such as multimorbidity and geriatric syndromes. First, I'm going to tell you a little bit about what I encounter here in the US and review some of the epidemiology of aging and non-communicable diseases that we're seeing in our patients who are living with HIV. From there, I'll shift the focus to a more global one, as the US is not the only setting. Is someone speaking? Okay, I'll keep going. I have a hard time seeing the chat when I'm presenting, so please just jump in and interrupt me if you need to grab me.
I'll let you know, Jessica, if
Okay.
something comes up.
Okay, thanks, Vivian. So from talking about the US, I'd like to shift my focus to a more global one, as what we're seeing in HIV and aging is not just happening here in the US, but is really happening in many places around the world. I'll introduce the research network that I work closely with, the Caribbean, Central America, and South American Network for HIV Epidemiology, and go into some of our group's recent work in the area of HIV and aging. And towards the end of the talk, I'll talk about some of the key challenges and opportunities in this field as we look ahead. And I'll close with describing the project that I'm working on with Vivian and Esper and others at USP, the longitudinal study of HIV and aging in Brazil, and how we hope through our study we can address some of these important knowledge gaps in the field. So first, starting out what we're seeing in the US. It's no secret that thanks to the success of antiretroviral therapy, adults living with HIV are living to older ages. And it's now the case that older adults with HIV are not the exception, but are rather the majority of the patients that we're seeing in our clinics here. Based upon data from 2018, the CDC estimated that of all people living with HIV in the US, 51% were over 50 years of age. Further, we know that this demographic shift of people living with HIV in the US is only going to continue to increase in the near future. These estimates predict the population of adults living with HIV in the US from the year 2006 through 2030. And it's estimated that by the year 2030, 1 in 5 adults living with HIV in the US will be over 65 years of age. While 1 in 6 new HIV diagnoses in the US are among adults over the age of 50, the largest contributor to this growing population of older adults with HIV is a result of the improved life expectancy thanks to antiretroviral therapy. These data are from a nationally representative consortium of adults living with HIV and in care in the US and Canada called the NA Accord. And in this study, they show the estimated number of years of life remaining from the age of 20, that's on the y-axis, and how that what that estimated life expectancy from age 20 is across 3 calendar eras among people living with HIV. So the earliest calendar era is the year 2000 to 2002, then 2003 to 2005, and then finally 2006 to 2007. And we can see that overall, from the early 2000s to the later 2000s, there was a steady increase in life expectancy. This increase in life expectancy generally occurred across key demographics and across CD4 count. However, important disparities are easily seen in these trends with respect to life expectancy, and key populations are still experiencing lower life expectancy overall. More recently, the same group updated their analysis, now looking at years past 2007, and found that unfortunately some of these disparities remain. Using data now from 2004 to 2007 in the lightest bars, but now adding the years of 2008 to 2011 in the middle toned bars, and then 2012 to 2015 in the darkest bars, they are showing now on the x-axis the added years of life expectancy estimated from age of 20 among people living with HIV. So now the axis has shifted a little bit. So starting with this top corner in the right, they looked at important disparities in life expectancy gains over time. So comparing Black and white men who have sex with men who are living with HIV, we see that there's been continued increases in life expectancy for both groups in the more recent years, shown in those horizontal bars. We also see, looking at the vertical bars, a narrowing in the racial disparity of life expectancy from the earlier years to the more recent years. This change in disparity is different than what we observe for individuals who identified injection drug use as their HIV acquisition risk factor. Again, while life expectancy has improved for individuals with and without a history of injection drug use, shown in the horizontal bars, the overall life expectancy gap between the two groups has actually remained stable and has not narrowed over time, highlighting the need for ongoing efforts to reduce these important disparities in lifespan. However, life expectancy and lifespan really only tell part of the story of aging with HIV. What about health span and years of healthy life? And this was an important study that was recently published using data from the Kaiser Permanente California and Mid-Atlantic research groups. Kaiser Permanente is a comprehensive healthcare network, and so they're including information about individuals in their closed healthcare network who are living with HIV and who do not have HIV to serve as controls. They matched on key demographics such as age, sex, race, medical center, and calendar year at the start of follow-up. And first, they looked at number of years expected of life from age 20. And then they also asked the question about the number of years expected to remain free of a major medical comorbidity. And those are shown in the top right. And for those in the room, because it may be slightly small to read, those major comorbidities include chronic liver disease, chronic kidney disease, lung disease, diabetes, cardiovascular disease, and cancer. So first, shown in the gray graph at the top, and consistent with the other studies, they've shown that over the calendar eras from 2000 through 2016, a narrowing in the life expectancy disparity between individuals in their healthcare system with and without HIV infection. And in fact, by the end of the study period, there was only a 3-year difference in expected life, calculated life expectancy between individuals with and without HIV. This is great news, and this is news that we can share with our patients. However, this pattern contrasted compared to what they saw for health span. So now looking in the orange bars, they calculated the number of years of life expected to remain free of a major medical comorbidity. And now, compared to the gray bars where we see a narrowing over time, we see that the individuals in their population living with HIV consistently experience fewer years without a major medical comorbidity. And in fact, that disparity in expected time free of major comorbidity has remained constant over time, with on average individuals living with HIV experiencing their first major medical comorbidity 16 years earlier than matched controls without HIV, all in the same healthcare system. They went on to look a little bit more specifically at the different comorbidities. And again, for folks in the room who this may be smaller to read, the top right box, actually, let me see if I can do a pointer here. Here we go. This top bar on the top box in the left here is showing liver disease. Panel B is kidney disease. The top right is lung disease. Then we have diabetes in D, cancer in E, and cardiovascular disease in F, showing years expected to remain free of these major comorbidities for individuals without HIV in the gray graph, and compared to those living with HIV in the orange graph. And again, you can see that this disparity in terms of relative years of life without these major comorbidities is fewer for those individuals living with HIV, suggesting that while their lifespan is improving and is reaching nearly the same expectations for individuals without HIV, they're aging with a higher burden of comorbidities, and their relative health span is actually much shorter. In the past few years, we have seen a spotlight on HIV and aging really turned up, as medical providers and the HIV research community are increasingly focused on caring for older adults. The impetus for this heightened focus really comes back to the voices and advocacy of adults living with HIV who are demanding attention to their complex needs to address these gaps in health span. They are raising awareness and demanding attention to improve health outcomes in older adults with HIV. Some of the key advocates include Jules Levin in New York City, Mark Thompson in London, and Benita Ray in Houston as examples. And all of these advocates have been featured speakers at some of our major HIV international conferences. At the opening plenary of CROI 2022, advocate Mr. Mark Thompson taught us. He noted that one thing that all of us who are here today share, we are all aging. We can't escape it. It's a process and a journey that is inevitable. But for those of us who have HIV, there are many added layers and dimensions to consider. It is not enough for us just to be grateful that we are still here. For us in research, we need to heed this call to better understand the determinants and possible points of intervention to improve quality of life and health with aging, not simply just adding years of life. So as the Kaiser study suggested, the excess burden of non-communicable diseases experienced by people living with HIV are a major determinant to their health span. Many studies have shown that adults with HIV are at increased risk for a number of non-communicable diseases compared to their peers. Adults living with HIV experience higher rates of cardiovascular disease, cancer, metabolic disorders, osteoporosis, renal disease, and liver disease. Additionally, we know that many of these conditions do not occur in isolation. This means that adults with HIV have higher rates of multimorbidity, or 2 or more of these chronic comorbidities, as shown on the graph on the right, which demonstrates data from a matched study of middle-aged adults with and without HIV, and showing that compared to individuals without HIV, those living with HIV experience a higher burden and an earlier onset of multimorbidity. Recently, the burden of NCDs and multimorbidity, the accumulation of NCDs, has become greater interest. And again, using data from the NA Accord in the US, they looked at the prevalence of multimorbidity on more than 22,000 people living with HIV from the years 2000 to 2009. They included individuals who had developed the aging-associated conditions of hypertension, type 2 diabetes, chronic kidney disease, hyperlipidemia, and end-stage liver disease, as well as non-AIDS-related cancers, and defined multimorbidity as having 2 or more of these conditions. So, not surprising, they observed an overall increase in the prevalence of 2, 3, 4, 5, or even 6 of these aging-related chronic medical conditions among people living with HIV going from the year 2000 to the year 2009. In fact, the prevalence of multimorbidity increased from 8% to 22% over this time period. What was a little bit surprising is that this increasing burden of multimorbidity was not simply driven by age. So they stratified and compared the prevalence of multimorbidity, comparing individuals included in the year 2000 versus those who were included in the year 2009. And you see that after you stratify by age, even comparing individuals of 50 to 59 or greater than 60 years of age, there was a greater burden of multimorbidity in the older adults in the more recent years compared to the older adults in the earlier years of the study period. Additionally, they identified other risk factors for multimorbidity, including identifying differences by race, HIV acquisition risk factor, BMI at the start of antiretroviral initiation. And in this study, they did not observe a difference in risk of multimorbidity by sex. Indeed, we've identified that there are a number of important risk factors for multimorbidity in adults living with HIV. Echoing observations for their association with NCDs more specifically, and building upon possible underlying biological mechanisms, a number of immunologic biomarkers in adults living with HIV have been associated with an increased risk of multimorbidity, including a history of a low CD4 nadir, low CD4-CD8 ratio, and measures of T cell activation and senescence. We also know that virologic biomarkers such as viremia and years of virus have been associated with risk of multimorbidity down the road, as well as factors such as females and history of depression. And to share a little bit about the results related to depression, this was a study that we conducted using data on more than 4,000 adults living with HIV who were cared for at our HIV clinic here in Vanderbilt. And we examined the association of a prevalent mood disorder and risk of future incident non-communicable diseases. Their first NCD, as shown in the highlighted box here, as well as developing 2 or more NCDs, 3 or more NCDs, or even a metabolic syndrome. And we found that even after adjusting for pertinent covariates, individuals with a history of depression carried an increased risk for developing these non-communicable diseases over time. And this particularly stood out for those conditions associated with the metabolic syndrome. However, multimorbidity from NCDs is not the only aging syndrome experienced by people living with HIV. And in fact, clinical studies have shown that older adults with HIV and those with multimorbidity in particular experience a higher risk of other geriatric syndromes, including risks of hospitalization, decreased quality of life, increased measures of polypharmacy or 5 or more daily medicines per day, increased healthcare utilization, as well as increased risk of cognitive decline and frailty. This growing aging-related morbidity among people living with HIV has been recognized for some time in the US, and I include on this slide some pictures of some individuals who are living with HIV and highlighting some of the aging-related morbidity that they reported in this article in New York Magazine, now almost more than 10 years ago. So I mentioned frailty, and so before I go much further, I first wanted to take a minute to talk about frailty. So frailty is often a state that we kind of think we know it when we see it, but I want to give you a little bit more of a definition of what we're talking about when we talk about frailty. So frailty, as defined by this nice review from The Lancet, is really a state of increased vulnerability to poor resolution of homeostasis after a stressor event. So thinking more pictorially, if we imagine 2 individual who's our green person on this top line, and another individual who's frail as the red line below them. So at baseline, before experiencing some type of minor illness, like a urinary tract infection or maybe pneumonia, both the non-frail individual at the top and the frail individual at the bottom may both appear fairly similar. They may both be functioning independently, and you couldn't tell on the surface that the individual in red actually carried a frailty phenotype. Where the frailty shows itself is in that stressor event. So both of these individuals experience the same stressor, a urinary tract infection, a pneumonia, a fall. But with that stressor event, the non-frail person experiences a dip in their functional ability, but then rapid improvement back up to their baseline and continuing on their way. The frail individual with that same insult experiences a much steeper loss in their functional ability. Maybe now they're actually more dependent. They need to spend some time in a skilled nursing facility, get extra physical therapy, start to use a walker, and they have a much slower and more gradual return to a level of independence. And perhaps they never make it all the way back up to the level of independence or functioning that they had before the event. So in a more straightforward way, I think of frailty as really the loss of the ability to bounce back. And again, we really see that in a stressor situation. So making this a little bit more complex, I want to tell you a little bit about the science behind frailty. And this diagram describes the cycle of frailty and the inputs that contribute to its development. So first, we start with an underlying disease, and perhaps that's something like HIV that's associated with senescent changes that ultimately result in a loss of muscle mass or sarcopenia. This loss of mass may impact strength, which we can measure by grip strength when we do the Freed Frailty Assessment, or exercise endurance, such as their fatigue. And this ultimately results in a slower performance and loss of endurance. This loss of power may ultimately then lead them to have decreased activity overall and become more sedentary, resulting in lower metabolic rate and decreased total energy expenditure. When combined with the loss of muscle mass, this decreased energy expenditure can result in loss of appetite and chronic undernutrition. And the decreased caloric consumption results in further weight loss. And again, you see the cycle of frailty, ultimately then leading to more loss of muscle and sarcopenia. Frailty is not a new concept in HIV, and adults living with HIV were first noted to have higher prevalence of frailty in the pre-ART era, demonstrating that they had higher rates of early frailty. However, even since the introduction of antiretroviral therapy, adults living with HIV continue to have higher rates of frailty and earlier onset of frailty compared to their matched peers. This data, now not all that recent, came from the MAX cohort, which is a longitudinal cohort of men with and without HIV. And you can see that the individuals living with HIV in the black diamonds experience an earlier prevalence of frailty starting at age 50 compared to their matched uninfected controls. And this difference in prevalence of frailty in adults living with HIV has also been replicated in international cohorts as well as cohorts of women with and without HIV. Importantly, frailty remains a critical predictor of mortality in people living with HIV. And these data are from the ALIVE cohort, individuals with and without HIV, all of whom have a history of injection drug use in the US, and they compared likelihood of death or overall survival, comparing individuals with and without HIV and with and without frailty. And you can see that those individuals that were both living with HIV and also defined to meet frailty criteria experienced a 7-fold increased risk of mortality compared to the group that had neither HIV nor frailty. So when thinking about the reasons of the high burden of non-communicable diseases, the earlier onset and higher rates of geriatric syndromes in adults living with HIV, there are a number of complex, interacting, but common etiologies that have been described in the literature to explain what we're seeing epidemiologically. We know that there are influences such as age and sex, family history, and BMI that drive these outcomes. But we also know that there are important contributions by co-infections and comorbidities, such as chronic hepatitis viruses, other comorbidities such as hypertension, and I've shown you some data about how depression can drive these outcomes. Further, we know that even with successful treatment with antiretroviral therapy, those measures of immune dysfunction and chronic inflammation remain important predictors of non-infectious outcomes in adults aging with HIV. We know that ART itself carries risks for some of these conditions, particularly our earlier drugs and exposure to the D drugs, protease inhibitors, and tenofovir all carry long-term effects on risk of cardiovascular disease, bone disease, kidney disease, and others. And importantly, what we still have much more to learn about is understanding the role of social determinants of health and how those impact these conditions and help us understand why individuals with HIV may be particularly vulnerable to aging-related morbidity. And now what we have in the background is the biologic processes of aging as this group is living longer and reaching older age. So indeed, this importance of HIV and aging-related morbidity that I've described in the US is not a US only consideration. And there's been calls for many years for an increasingly global focus of understanding HIV and aging. In this nice editorial that was, again, published almost 10 years ago, colleagues called on the importance of looking at NCDs and aging-related morbidity, not just in high-income settings, but in low and middle-income settings where the majority of people living with HIV in the world live. They acknowledge that differences in local, environmental, genetics, and HIV treatments, as well as co-infections, may all affect the prevalence and determinants of these non-communicable diseases and aging morbidity. And further, they noted that the public health infrastructure that's been built over the years to care for individuals with HIV may be able to be leveraged to improve the diagnosis treatment of some of these non-infectious conditions, particularly in resource-limited settings. One of these important reasons for understanding non-communicable diseases in low and middle income countries more generally is that we know that the global demographics are rapidly shifting. So in many countries, we are seeing improvement in life expectancy and a reduction in birth rates that are fundamentally changing the population structures around the world. And this graph highlights the tremendous change in population demographics that are currently occurring in Latin America. At the bottom, you can see the structure of high-income countries with the bulge of the population occurring in middle-aged adults and the projected increase in older adults in the general population looking ahead to the year 2050. The top shows the change that's been observed that's projected in countries in Latin America, comparing the population in 2010, which was disproportionately younger and under the age of 40, to now a projected middle-age bulge coming in the year 2050. So to better answer the call to understand HIV and aging from a more global perspective, I first want to introduce the research consortium that's near to my heart, the Caribbean, Central America, and South America Network for HIV Epidemiology, or CASANET. Like NA Accord, which I introduced earlier on, CASANET is one of the 7 global regions that make up the international epidemiologic databases to evaluate AIDS, or IDEA. And these consortiums include and harmonize data from adults and children living with HIV and receiving routine clinical care from around the globe. Within CASANET, our focus is to understand the HIV pandemic here in Latin America and the Caribbean. And our data includes observational data on more than 60,000 adults and children living with HIV in clinical sites in Mexico, Haiti, Honduras, Peru, Chile, Brazil, and Argentina. And Vanderbilt serves as the data coordinating center for CASANET. In recent years, our group is becoming increasingly interested in older adults living with HIV in our region. And as in high income settings, people with HIV in Latin America have experienced increased lifespans as a result of ART. This recent study from our group, which was led by a mentee of mine at Vanderbilt, used life tables to calculate life expectancy from age 20 among adults who were enrolled at CASANET sites and receiving antiretroviral therapy from the year 2003 to 2017. We examined life expectancy trends across 3 calendar eras and used a regression model to account for likelihood of loss to follow-up. We also looked across trends in key demographics and clinical factors at ART initiation. And in total, we included more than 30,000 individuals. As I show on this slide, a large proportion of our CASANET cohort comes from our clinical sites in Haiti. And given the differences in the history of the HIV pandemic there compared to our other sites, we stratified by the analyses by Haiti and the all other sites. So what did we find? Overall, we observed that for both all of the sites as well as the Haiti site, which is shown in the red line, and all other sites shown in the blue line, there's been a steady increase in life expectancy among adults started on antiretroviral therapy across the calendar eras. And using the most recent UN data from 2018, we show that those overall life expectancies among individuals living with HIV are starting to approach or nearing those of the general population. Importantly, we observed increases in life expectancy across both men and women in Haiti, which is shown in this top bar here. So women shown in blue and men shown in red, as well as improvements in life expectancy among the other sites by HIV acquisition risk groups, including heterosexual women shown in blue, men who have sex with men shown in red. Green is heterosexual men, and those individuals with injection drug use, other or unknown HIV acquisition risk factors shown in purple. We also looked at improvements in life expectancy by CD4 count at ART initiation. This first column here are the data from Haiti, and then the column to the left are the all other sites. This top panel is looking at individuals who started ART with a CD4 count above 200 in the blue line, compared to those who started ART with a CD4 count less than 200, shown in the red line. We also looked at life expectancy gains by whether or not individuals had a history of tuberculosis at the time of starting antiretroviral therapy. no TB shown in the blue lines, history of TB shown in the red lines, and finally looked at differences by education, upper secondary education or higher shown in the blue line, lower education shown in the red line. These improvements in life expectancy really confirmed what our collaborators were seeing in their clinics. And highlighted in this nice study by colleagues from Mexico City, they noted that among all CASINet clinical sites, we've observed an overall increase in the proportion of adults over the age of 50, from 8% in the year 2000 to now 24% as of data from 2015. While the rate of this demographic shift has been steady across calendar eras, we've also seen some of the steepest slopes for aging occurring in some of our lower income settings, such as the purple line, showing the relative increase in the proportion of adults over the age of 50 from our site in Honduras. Looking more closely at the older adults in our cohort, Dr. Belanzaran, also from the group in Mexico City, led a study looking at the prevalence of comorbidities in this population. And including more than 3,000 adults in the cohort over the age of 50, he and his team stratified this population by those who had enrolled in care before the age of 50 versus those who entered care after the age of 50, and looked at the annual prevalence of one or two or more key comorbidities. Overall, the adults that enrolled in the clinic after the age of 50 were followed for fewer years than those who had enrolled before the age of 50. So looking at the graphs overall, they observed that the adults who enrolled at an older age had a fewer number of prevalent comorbidities at age 50 compared to those who had entered care before the age of 50. It could be that those individuals who entered care before the age of 50 had a longer time living with HIV and to develop the effects of HIV and its treatments, or it could be differences in healthcare access and opportunities for diagnosis of these comorbidities. However, when they looked at the end of the study follow-up, regardless of whether individuals started care before the age of 50 or after the age of 50, both groups had similar prevalence of one comorbidity or two or more comorbidities, showing the high burden of multimorbidity in this population. So our experiences studying NCDs and other aging-related outcomes in Latin America has brought to the forefront some important challenges in observational research. And while some of these may be marked in settings with less resources, they are present in high-income settings as well. And as we observed in our life expectancy period, there are some key differences across countries and regions with respect to underlying life expectancy and ART policy history. Studying non-infectious endpoints like NCDs can also be challenging, where differences in medical records and systems, as well as fewer opportunities for screening and diagnosis, may result in missing or misclassification of NCDs, reminding us that the absence of evidence is not the evidence of absence. So to address some of those challenges, our group has decided to reduce some of that heterogeneity by focusing on Brazil, which is a very important setting for understanding HIV and aging. As you all know, Brazil was one of the first middle-income countries to make ART available free of charge for individuals living with HIV and was an early adopter of the Treat All era. The largest country in Latin America, it has an estimated nearly 1 million people living with HIV. And through its public health program providing antiretroviral therapy, we see that rates of HIV diagnosis among those estimated to be living with HIV, those started on ART, and those achieving virologic suppression are nearly 90% across the country. With that background, back in 2014, CASINet received a supplement from the NIH to study NCD outcomes among individuals who had started antiretroviral therapy in Brazil. And we partnered with COARTI Brazil, which was an existing multi-site observational study funded by the Brazilian Ministry of Health to examine clinical outcomes following antiretroviral therapy start. With our CASINet collaboration, the COARTI Brazil project worked to validate and collect NCD diagnoses among individuals starting ART at 7 clinical sites from 6 cities within the COARTI study. Clinical staff abstracted NCD diagnoses from medical records, which were then internally validated and underwent external data audits for data quality assessment. Overall, we observed that among this large cohort, the proportion of adults over the age of 50 during the study period steadily increased from about 9% in 2003 to nearly 24% by the year 2014. In parallel to this aging of the cohort, we saw an increasing prevalence of one or more or two or more NCDs in the cohort, again, highlighting that growing burden of multimorbidity. Overall, we found that women had a higher cumulative incidence of multimorbidity compared to men. And in our multivariable analyses, looking at other risk factors for multimorbidity, we found that those individuals who started ART with a low CD4 cell count, as well as those who started ART in more recent years, had higher rates of multimorbidity over time. In addition, we know that there are some other important research challenges and questions that are needed for observational research in HIV in aging. One of these important challenges is to identify and to measure some of the unique risk factors and the influence of local exposures on aging-related morbidity in adults living with HIV. And this means we need to address challenges in capture of some of the social and behavioral factors that can contribute to these outcomes. Additionally, we've recognized that just relying on medical records isn't enough because those sources tend to not have information about quality of life, physical function, cognition, and some of the geriatric syndromes that we know are important not only for lifespan, but also important for health span. The importance of unique and complex risk factors to understanding HIV and aging is really critical for moving the field forward, particularly when we think about how social and structural determinants impact health and longevity. Further, we need to think about these social determinants of health, not just at a single time point, but we need to apply life course methodology to our studies, recognizing that the exposures and experiences early in life often have important consequences downstream. Observational research that rely upon use of medical records have important strengths with regards to documentation of validated NCDs, but they're often limited in what we can gather about the important interpersonal, community, and policy factors, all of which significantly affect exposures that contribute to risk of aging-related morbidity. Further, we know that these social and ecologic exposures are not fixed, and future studies need to work to help elucidate how time-varying and cumulative exposure to these social determinants of health contribute to aging outcomes in adults living with HIV. Another challenge in HIV and aging is to fully incorporate the lessons of geroscience into our research. The inner ring of this circle describes some of the principal cellular mechanisms of aging, including inflammation, senescence, cellular metabolism, and others. The principles of geroscience teach us that these cellular processes are important because they are common not just to one disease of aging, but are common to multiple diseases of aging and NCDs, as shown in the middle circle. They also can lead to more systemic processes of aging, such as frailty and functional impairment, shown in the outer ring. In infectious diseases, we need to understand mechanistically not only how HIV, but other chronic pathogens contribute to these cellular processes, and recognizing that these infections, not just HIV, affect lifespan and health span. So I'm really excited to tell you a little bit about the recent grant that we and our team at USP and others received from the National Institutes on Aging to work to address some of these challenges. Through the longitudinal study of HIV and aging in Brazil, or ALEA, we will build a prospective cohort of older adults living with HIV in Brazil to collect longitudinal data on comorbidities, particularly cardiovascular disease, as well as geriatric syndromes. And we'll collect systematic information about social support, socioeconomic factors, and behavioral data to investigate both the ecological and the immunologic inputs that lead to aging-related morbidity. Overall, our overarching objectives of the study include to define the burden of how some of these endemic co-infections, as well as the unique socioeconomic factors, are associated with NCDs and geriatric syndromes. and then longitudinally to understand the clinical predictors of geriatric syndromes in older adults living with HIV. And our final aim is a translational one to look at these inputs, including co-infections and other social determinants of health on inflammatory pathways known to be associated with some of these aging outcomes. Shown graphically, ALEA is now finishing up enrollment of nearly 700 adults all over the age of 50 and living with HIV, receiving care from 4 clinical sites across Brazil. All the participants undergo a comprehensive baseline visit and then are followed longitudinally to look at incident cardiovascular disease, geriatric syndromes, and adverse clinical outcomes such as hospitalizations and death. This has really been a truly wonderful international collaboration, drawing upon clinical and laboratory expertise from institutions throughout Brazil, as well as US, Canada, and Germany. Just to tell you a little bit about our cohort, and I know I'm wrapping up on time. So far, we've enrolled over 500 individuals. And as I show in this table here, we really have a rich and diverse cohort. We're purposefully trying to enroll individuals over the age of 65. You can see that about a third of our cohort are women, but we also have a number of individuals who are transgender or gender nonconforming. The overall average age in the cohort is 60 years, but we have individuals in our cohort well into their 80s. And we have a diversity of racial and ethnic background, as well as a diversity with respect to education and working status. This is really exciting, and we're now beginning to look to analyze some of our baseline data and to start our translational work. And I hope in the future I can return to USB to give you an update on some of our results. So I want to wrap up and just conclude. I hope that I've impressed upon you that older adults with HIV are increasingly becoming the majority of the adults with HIV we're caring for in our clinics. And this is happening around the globe. While we've made great improvements in health span, we have a lot more to do to improve. While we've made improvement in lifespan, we have a lot more to do to improve the health span of aging adults with HIV. The burden and causes of NCDs and geriatric syndromes in adults with HIV are complex. But our understanding of the roles of social determinants of health and the mechanisms of geroscience that contribute to these morbidities are incomplete, and we hope to address some of those gaps with our new study. More research is needed for the epidemiology of aging-related morbidity really around the world, and we have to address some of the limitations that are inherent to this research, as well as recognizing unique opportunities. And I hope I've shown you that collaborative research around the globe really can lay the groundwork for this important work to be done. I'd like to recognize our collaborators with ALEA and in particular, Vivian and Esper at USB. And really they are just part of a great team that we've worked with here at USB. I show a picture here from our recent visit with Vivi joining us on the screen, but many thanks. And it's been such a fantastic experience working with all of you. So with that, I'll say thanks from Nashville, and would love to answer any questions if time permits. Thank you again for this opportunity.
Thanks, Jessica. It was great, comprehensive lecture on this topic, which gives us a lot of information and important points. I really like the concept of health span as opposed to lifespan. And for those of us who are interested in the business of making our patients live not only longer, but also better, that is really important. Thank you so much for that. Angela, do we have any questions from the audience on YouTube?
No, on YouTube, no. Pessoal, alguém aqui gostaria de fazer alguma pergunta para a Jéssica, que nos brindou com uma excelente palestra, né, que foi absolutamente completa, né, sobre envelhecimento. Jessica, thank you very much for your conference. It was very, very complete and about the being old with HIV and the difference between who has HIV and who hasn't. Eu não sei se alguém teria alguma pergunta nesse momento para para fazer, se ficou alguma dúvida, se querem, né, saber alguma coisa do ELEA, do estudo que está acontecendo. Eu sei que a gente tem tanto médicos, né, que atuam aqui com HIV, quanto residentes. Eu acho que é um momento interessante para a gente falar também que se alguém tiver interesse em se juntar ao time do ELEA ou estudar envelhecimento em população com HIV, existe essa possibilidade, né, tanto do ponto de vista clínico, quanto do ponto de vista de ciência básica na bancada, né, junto com a equipe do laboratório. Tá? Vivi, está com você.
Jessica, thank you so much once again. Jessica's lecture actually gives us perfect background to understand why we still need to cure HIV, which is the topic of our next talk with Melanie Ott. Jessica, thanks again so much. Melanie, I'll give you the floor.
Yes, thank you very much, Vivian, and hello, everybody. Also, Jessica, thank you so much for that wonderful lecture and synopsis. I think HIV and aging is such an important aspect in motivating us to do what we're doing and what I'm going to talk about in the next 40-50 minutes. My name is Melanie Ott, as Vivian already said. I'm in San Francisco. I'm a virologist. I lead the Gladstone Institute of Virology here in San Francisco. We are closely affiliated with UCSF, but we are an independent research institution. And I'm going to talk about the HOPE Collaboratory, which stands for HIV Obstruction by Programmed Epigenetics. And this is a large collaboratory that I co-lead together with my co-PIs, Lish Lovu and Susana Valente. So the main goal of HOPE is to block, lock, and excise HIV. And I just wanted to start with just defining exactly what we mean with this. Blocking means we want to inhibit HIV transcription, which I think is very understandable. But we not only want to block it while we give the drug, we also want to lock the HIV, the provirus, in a state of permanent silencing. That means we want to keep the virus blocked without therapy. And we want to eventually excise or permanently inactivate the proviral DNA in the genome. And that means that by using epigenomic and genomic approaches, we want to permanently inactivate HIV. I did not include any slides on latency and showing that this is basically a major problem, but just wanted to sort of indicate that the reason why we're still talking about HIV and a cure is that antiretrovirals do not provide this cure program, and that some transcriptionally inactive viruses persist in people even if they're fully virally suppressed. And that's what we're going after. We're going after these latent proviruses. And we think that this block, lock, excise strategy is actually a very natural way to deal with this latent reservoir. Because we have a blueprint of other retroviruses in our genome that have gone down that path in precisely that way. However, that was not therapeutically engineered. These endogenous retroviruses, which make up a considerable amount of our genome, have invaded our genome millions of years ago, but they have decayed over time into not dangerous and non-pathogenic viruses. And we want to use this blueprint to accelerate HIV's natural path to become such an endogenous retrovirus. We are a international multi-site collaboratory. We're led by San Francisco, New York, and Florida here. But we have 16 sites in 6 countries, and most importantly, of course, we have Esper and Vivian's group here included in the HOPE collaboratory. Now, what is our scientific program? So how are we, what are we focusing on? We are we're using a very molecular approach to come to curing this remaining, persisting reservoir that we find in people living with HIV, but even fully virologically suppressed. We divide the stages of infection into 3 parts. One is the active part, and I'm showing you here down below that the promoter and what is going on at the HIV provirus in the T cell or other cells when there's active transcription, and that is actually the state of the provirus in most cases, because the virus integrates into active genes and it has a powerful engine itself, which is called the TUT protein, that drives active replication. However, we know that in a small percentage, the virus is actually becoming latent. That means that it does not produce any more viruses. It is sort of dormant in the cells. However, this latent state is a very fragile state because it can immediately go back and forth between the active state. And this is because there's still a little bit of activity happening on the promoter, which basically prevents the locus here from being silenced. So the third state is actually the silent state, which you might notice here is irreversible. And we have hypothesized that this state would exist, but now with new literature in the last year by Matthias Lichterfeld's group, where he was able to really characterize the latently infected cells or the remaining infected cells in people living with HIV, we know that this silent state exists where we don't have any more activity at the promoter. And we have basically epigenetic, but also genomic silencing with DNA methylation, really silencing transcription in this loci. Now, our goal is to use all of these stages for genomic interventions like CRISPR or other gene therapy interventions that I will talk about later, to actually excise the provirus, or at least inactivate the provirus at the genomic, at the DNA level, such that it is irreversibly inactivated. And I will go through these stages one by one. So the active stage is, as I said, characterized by a highly productive transcriptional program, and that is driven by a small protein by the virus, which is called the TUT protein, that binds to RNA. It basically binds to the transcripts, the viral transcripts that are forming by the action of the human RNA polymerase II. And TUT recruits a powerful host factor, which is called the super elongation factor, or a SEC, to this beginning of transcription, and that leads to an incredible uptick in transcriptional efficiency, especially in completing the transcript and having complete replication competent transcripts. Now, we in HOPE developing measures to target both the TUT, but also the host factors, which is here in this case, the superelongation complex. And I will talk more about this triad of TUT, superelongation factor, and TAR, which is really critical to potentially silence or block this step. Now, the latent step I told you is sort of pseudo-silence because the virus is dormant. There's nothing coming out of the cell. But we know that there's still quite a bit of activity in the way that the polymerase is sitting on the promoter constitutively. It's so-called in a paused state. And actually, HIV was the prominent condition where a paused polymerase was identified. And so this pause polymerase continuously makes little snippets of the transcript, which is enough to form the element that recruits the TUT protein, which is TAR, but it is not elongation competent because TUT is not being produced and the superelongation complex is not recruited. However, the presence of this polymerase keeps the locus open and prevents it from reforming so that it could actually become silenced. So we are interested in really targeting this state by targeting the polymerase that is sitting here, but also recruiting specific repressors that are counterbalancing the basal activity that the promoter still maintains. And if that is, successful, we think we get rid of the, of this remaining activity here. We get marks on the histones and marks on the DNA which silence, gene loci, and that can lead to a more permanent or a more robust or durable state of latency, which we call the deep state of latency, or we could call it silenced. And the important part is that it's very hard to re-go back into the active state from this state. And again, this is not yet sort of a permanent inactivation. So in order to achieve this, I think there's a final coup. And this is that we would like to use modern genomic tools to now remove or alter the HIV genome. And we want to do this very gentle and very targeted. And I will tell you in a minute about how we're doing this. And again, we don't have to do this in the silent state, which is usually a little bit complicated because of all the modifications here. We can do it in any state, the active, the latent, or the silenced. And then we would have basically nothing left or a permanently damaged provirus left. So let me tell you about how the program is structured. It's actually structured in three research foci. The first one is basically basic biology. And here we would like to define the state of HIV silencing by targeting specific host and viral factors. This RF1 is co-led by Susanna and Doug Nixon. But we have great members in here that contribute, specifically also Cedric Fechot, a world expert in endogenous retroviruses. You'll remember we want to learn from those to actually apply it to HIV. We also have Eric Verdin here, who is a pioneer in HIV chromatin research, but also is currently leading the Buck Institute of Human Aging, which is very much in line with what we just heard and is very interested in the molecular basis of HIV and aging. So here in RF1, the hypothesis is that HIV transcription and the chromatin structure offer unique targets for silencing. And we have 4 goals here. The first one is that we identify really new repressors and regulators that can induce proviral silencing, and we can then use the Cas9, CRISPR-Cas9 system to actually specifically recruit them to HIV and study exactly what they are doing. We want to learn more about endogenous retroviruses. They have, unique features. They have the zinc fingers that are, have evolved in us to actually specifically silence these retroviral elements. And we would like to know how we can harness this for HIV. And turns out that retroviruses, endogenous retroviruses are very heterogeneous. We're doing a lot of sequencing and classification work to really identify those that we want to learn from and those that we don't want to learn from. The third goal is really to understand better the role of TUT. I told you TUT is a really important transcriptional activator. We know from Susanna's work that if we target TUT therapeutically, we can actually achieve a state of deep latency. So TUT seems to be a really crucial linchpin between not only active and latent, but actually also active, latent, and silenced. And that's what we are interested here in this aim. And then, of course, we would like to know from cells that we have silenced, either artificially or that are naturally silenced over time. We want to learn by omic technologies how the state of silencing is actually characterized and what we need to achieve to actually go into the state of silencing. Showing you a few snippets here, partially also from my own lab. Si-Chon Lee, is a senior scientist in the lab. He has done enormous work on using CRISPR screens, so knocking down human factors and then identifying what they do to HIV transcription, and identified by doing this either individually or in combined screens, where he's not only knocking down one factor, but two factors, finding out that there is actually networks of factors then, and some of them previously undisclosed or unknown, that are very important for transcription, for HIV transcription. And especially these three factors were newly identified in a synergy screen with each other. And that work has led to the identification that there is actually a super repressor complex sitting at the HIV promoter in the latent state, that is composed of this BAF complex or the NSL complex, which is actually part of a complex called MOF. It has an acetyltransferase activity. BAFs are really chromatin remodelers, so they really, use the chromatin structure and bend it and change it. And also a very important protein called BRD4 that the field has worked very extensively on, and we have also extensively worked on. So these all interact, and they also work synergistically to at least block HIV transcription. And we are currently finding out whether it locks and silences also. But here, if you use cells from people living with HIV and you overexpress some of these factors like BCL7C or cancel2 coming from this new complex here, you can see that after reactivation of HIV transcription, this is diminished just by simply overexpressing in some of these cells these factors. And so we are looking much more closely now how we can use the knowledge of the individual parts of this complex to actually recruit it to HIV and see how we can silence it. A second story is the opposite, where we actually look at activators that are important for HIV transcription, in this case also coming from the host. This is done by another senior scientist in the lab, Daniela Böhm. She works on this methyltransferase enzymes, which are enzymes that confer methyl groups onto histones or non-histone proteins. And we have worked on the SMIT, a specific one, the SMIT family here, which is a unique family because it has this MYND domain interspersed with its methyltransferase domain. And we have worked in the past on SMIT2, which is actually a repressor of HIV, and now have a paper just published on SMIT5, which is a very powerful activator. SMIT5 is very little known, but it is very highly expressed in CD4 positive T cells. That's why we're very interested in it. And when we express it just by itself or together with TUT and the HIV promoter, we have about 100-fold activation of that promoter with this protein. So it's a very powerful activator of HIV transcription. We know it's a methyltransferase. We looked at what it methylates. It methylates a whole bunch of histones, which are all shown here. They're numbered here. But there's still a very big unclarity what histone is actually the natural target of SMIT5. And there's a lot of discussion about it. But what we found is that actually SMIT5 methylates the TUT protein enormously. And when we published our paper, we got about 10 requests from labs that have nothing to do with HIV to get this peptide, just to start, finding better ways to study SMIT5 and find potentially inhibitors for it. Now, SMIT5 methylates TUT. What about TUT? What is TUT doing to SMIT5? When we did some expression studies and co-IP studies here, we could see that the SMIT5 protein here was expressed differently if it was expressed without TUT or it was expressed with TUT. And actually, we had a lot more SMID5 when there was TAT co-expressed, and also this SMID5 also interacted directly with it, or interacted with the TAT protein. So there was an effect on TAT on SMID5 expression. We determined that this was post-transcriptionally. It's likely a stabilization effect. And we have identified a protein, which is a USP11, which interacts with TAT, that is actually responsible for that stabilization, because when we knock it down, that's the upper band here, you see that TAT is not any longer stabilizing the SMID5 shown here. And so what we currently working on is that the SMID5 has actually a very intimate relationship with TAT and the HIV promoter, that it is usually very lowly expressed in T cells. However, the TAT protein massively stabilizes it, and that leads to a co-activation of the HIV promoter together with TAT. And we think that USP11 plays a big role, and we're currently therapeutically targeting both SMID5 and USP11 to see how we can, whether this would break the TAT cycle sufficiently to actually drive the virus into silencing. Now, RF2 is our more applied RF. It's actually specifically, looking at how can we prevent rebound after we stop ART. And as you recall, we want to not have any more rebound after we stop ART, because we will apply some silencing, therapeutics there, so that the virus is not coming back, because it's not anymore in that labile state that it can reactivate. So we would like to develop the next generation HIV silencing approaches that prevent rebound in individuals. This is co-directed by Lish and Nadia Rowan. She is a senior investigator at Gladstone, has a lot of fun people as members, especially, of course, also ESPER, but this is really a group that is, bringing together very clinical expertise, but also very basic expertise. So here the hypothesis is that viral rebound is suppressed with silencing promoting agent after ART is stopped. And we have several goals again. The first one is to really look at candidate silencing promoting agents that are currently already in the clinic. This is where the clinical part is really very important, and the different cohorts that we have around the world. Because there are already SPAs that are in the clinic currently, and one of them is spironolactone, which is a drug that is an aldosterone antagonist, and is actually used widely for hypertension, but also for gender changes. And so specifically, Brazil is a very important partner here to help us identify or look in people that have HIV, but also take spironolactone for other conditions, to then see what is the state of the provirus in these people, and is there a silencing or a silencing effect of the drug on HIV. So this is a very big part of RF2. We also looking to develop novel silencing promoting agents, and I will show you a few of them. TAT is a major target, since we know already in studies from Susanna that inhibiting TAT makes the virus silent. And the third one is really to use sequence-specific silencing promoting agents by using either the targeting activity of the Cas9 to just see whether we can selectively identify what these silencing domains are that we identify in the RF1 research focus, to see what exactly does it do to HIV, and what and how could it be potentially therapeutically used. And I tell you a little bit more about this in RF3 also. So just to let you know, spironolactone is a powerful inhibitor of HIV transcription, has been identified in several screens previously. but is also a commonly used drug. And so we're working very hard on it. I'm just showing you a little snippet of data here from Lish's lab, where they have specifically looked at microglia cells that carry a latent HIV provirus that has a green fluorescent protein, which is usually not expressed. However, if you reactivate the cells, you can actually see a lot of green because the virus becomes active, and this is done with TNF-alpha. Now, if you treat the cell with spironolactone, you suppress this very powerfully, and you see this over time here in this red curve. If you use eplerenone, which is a similar drug but has no HIV suppressant activity, you don't see this suppression. So really to show that the powerful effect of spironolactone in vitro, and the goal is really now in these international efforts, including Brazil, to see whether it also has an effect in vivo. The other target I told you is the TAT-TAR and superelongation complex axis, and this is here shown with cyclin T1 and EFF4, because the complex is really a big complex. This is a senior researcher in my lab, Ursula Schulze-Garnen, who is a structural biologist and has really done over the years enormous work in solving this TAT-TAR interaction and how the superelongation complex with its several components, cyclin T1 and EFF4, is actually important. And you see the TAR structure, which is the RNA structure that forms at the beginning of every transcript. You see how the TAT protein, which is tiny, wraps itself around this, makes contact with that TAR structure only when it's embedded into that superelongation complex. And you see how the other superelongation complex factors like cyclin T1 and EFF4 are also important for this interaction here. And so this TAT-TAR axis has really been targeted at multiple levels at HOPE. I think classically they are developed competitors of the TAT-ARM, which is the part of TAT that actually interacts with the TAR element to inhibit this binding to TAR. One of them is a molecule called DCA that has been used in the past, but is incredibly difficult to synthesize. Others are that molecules that would directly impede or destroy the TAR three-dimensional structure. There is inhibitors of the TAR binding to the complex, to the whole complex here, including the cyclin T1 and the others. And there is a new approach that I will show you a few data about, where you can actually use degraders of the TAT protein, specific protarchs or molecular glues that lead to the degradation of that complex, which is something that Susanna is working on. And we're also working on genomic approaches to use CRISPR to modify this TAR sequence. And so here is a snippet of Susanna's work. This is the TAT protein. She has identified quite a few molecules, new and known, that actually lead to the rapid degradation of the TAT protein. And this, when you use these compounds now, you can use them to really decrease viral transcription tremendously. And importantly, when you reactivate the cells after 12 days of treatment with this, you can see that they are still inhibited, also they're not anymore applying the drugs at that time. And so this is the type of experiment that we're using to test the blocking effect, to see whether this effect would go actually beyond just the immediate intake of these viruses. And this is where the whole concept comes in, that if we fundamentally change the structure of the promoter, we can actually lead to permanent changes in the chromatin structure so that we don't have to continuously drug. The other one is that is what Ursula is currently doing. She has basically isolated all the different components that are critical, the TAR element, the TAT, and the cyclin T1 and AFF4, has put this into an assay and has then just by proximity found out whether the complex is formed or not formed, and is screening for and has done multiple screens with this assay to see whether there are compounds that would directly interfere with the formation of this critical complex that is at the heart of HIV transcription. And she has identified several compounds that she's currently following up, but we also have just engaged in a contract with NIH in Washington to use this assay to screen massively more libraries in order to get better hits. And so that brings me to the third research focus, which is RF3, where we're really going a little bit more exotic here and really using the current gene therapy methods that are out there and that are becoming more and more mainstream to disable the HIV provirus by targeted genome engineering. And this is co-led by Priti Kumar from Yale, together with myself. And we have also great people in the group here, especially also Niren Murthy, who is an expert, has nothing to do with HIV, or little, now he's an expert in HIV also, but is actually a bioengineer that develops delivery mechanisms of CRISPR and other enzymes. He's at the University of Berkeley, Karl Berkeley. But we also have a group in Germany with Ulrike Lange, who invented, together with Jan Chemnitz, an alternative excision module that I will tell you in a minute that is specific for HIV. So here the hypothesis is that in vivo delivery of genome engineering therapeutics permanently inactivates HIV without genotoxicity. And I think the key is here on in vivo delivery. So we don't want to isolate the T cells, treat them, and then put them back into people. And we also would like not to induce any genotoxicity. And there's a lot of the genotoxicity, because when you use, for example, Cas9 just straight ahead and have it excise with specific guide RNA, the DNA, it induces a considerable amount of double-stranded DNA damage, which is called genotoxicity, which is highly inflammatory and can cause problems. And so we're trying to do this in a more gentle way, which is double-strand break-free. We're using this enzyme called BREC1, which is actually interesting because it is a Cre recombinase analog, but it's highly trained on the HIV promoter, recognizes all subtypes, and is very efficient in excising the provirus without genotoxicity. We're also looking at not only Cas9, but dead Cas9, catalytically inactive Cas9, that is fused to other modalities in order to, modify the genome, especially, for example, cytidine or adenosine deaminases, which can then base edit very gently the specific bases in the provirus without, making major deletions or cutting, events here. And then we're also working on peptide nucleic acids, but this is, I would say, the top two are currently our main focus. And I mean, the important part is, how do you deliver this? I think everything in gene therapy is delivery. And as I said, we need a highly effective delivery because we would like to use this as an injection module, not like a cellular therapy activity here, because otherwise it will get way too expensive and complicated. So we're working currently with virus-like particles, which is basically an inactivated HIV virus, but we are incorporating the module here as a protein into the virion, so that it gets delivered, transiently expressed, and then it's basically eliminated. So there's no integration of anything and permanent expression. It's a very transient activity here. We also using lipid nanoparticles, like in the SARS-CoV-2 vaccine, very much, or we're using protein peptide conjugates, and we're making a lot of progress and really putting specific targeting modules on all of those. So here's a little snippet from Britty's work. She's really a pioneer and an expert in humanized mouse models. And she has worked very early on in using CRISPR against, or it's Cas9 against CCR5, the co-receptor of HIV, and has shown here in this experiment that you can package CRISPR with guide RNAs into VLPs. I showed you this is basically into an HIV virus-like particle. You can inject this into the mouse, and then you can look for CD4 T cell death after infection. And you can see that that actually the VLPs, which are specifically targeted to CD7 expressing T cells, are effective in protecting CD4 cells from dying. And this paper is going to come out, very exciting data, especially also long-term data, where this is shown that this is actually highly protective and persists. The other approach that we're taking, I told you already, is a parallel approach to the CRISPR approach. It's called, we were focusing on a protein called BRCA1, or broad range recombinase 1, which is a protein that is similar to a Cre-like protein that is known in biology, where you don't need a guide RNA, and the enzyme is by itself recognizing only certain sequences. And this was selected to recognize the LTR sequences of HIV and can lead to this, excision process. Is very highly effective, has been shown by the German group in 2016, and there's a clinical trial in Germany going on with this enzyme currently. We like this enzyme because the sequence that it recognizes is highly conserved. It is very little immunogenic, so we can actually inject it multiple times if necessary. We don't need a guide RNA because it's trained to only recognize HIV, and there's very little off-target effects that have been reported. So and it doesn't make any double-stranded cuts. So it is actually a gentler way of excising. So this is something that's going on currently, and that is being pursued, and we using different packaging and delivery methods to do this, but it's mainly pursued in Germany. One thing that we are doing is using the targeting activity, the specificity of that BRCA1 recombinase for HIV, to use it in fusion proteins, similar to the dead CRISPR, where you inactivate the CRISPR activity and then fuse it to other things just to target it to a specific locus. We can use the BRCA1 now to actually inactivate it and then fuse it to other modules so that it makes specific silencing effects. And Jonathan Weissman's lab in 2021 published this CRISPR-off technology, where you use the dead Cas9 fused to a GRAP domain, which is what recruits repressors to silence, for example, endogenous retroviruses, but also specifically fuse the DNA methyltransferases with this to not only have histone methylation, but also DNA methylation, which I said is for us the hallmark of silencing. So we basically reenacted this cassette with BRCA1. Why would we do this? Why would we not go with this? This is a gigantic construct, and nobody can get it into T cells. Plus, you need a guide RNA that you also express in order to target it. So it's a very big construct. BRCA is about 1/3 of Cas9, and it is also, it doesn't require a guide RNA. So we think that it is a very valid alternative for us because it is so specific for HIV. And so the first thing that Ulrike in Germany did is she generated a point mutant in BRCA1 to kill the recombinase activity, and showed that it still binds to the HIV LTR, both by chip and here also functionally, because when we just simply overexpress the dead BRCA1, it competes with the polymerase to silence HIV. But it does not excise the integrated HIV because when you overexpress it over a longer time, you don't see any changes here in the cell. So we use this dead BRCA1 cassette and replaced it in the CRISPR off cassette with, for the dead CRISPR, and looked whether that would do anything. And this is a work by Francisco in my lab. And you can see when he overexpresses GFP, there is nothing. But if he overexpresses the BRCA off, it has actually pretty powerful silencing. But there's similar effects if you do this transiently, when you do this just with a dead BRCA1 without the silencing machinery attached to it, as I just showed you. However, when we do this over longer time, we see that only the BRCA off, which has the methylation modules associated with it, actually suppress HIV transcription over a long time, while the dead BRCA1 alone is not doing this. So we're currently doing a lot of experiments with this just to show what is actually going on and how robust is this suppression over a long time. But this ends the scientific part of the talk. I want to tell you that our fourth pillar, which is absolutely critical, is our community work at Hope. This is led by Patricia Defestreur, who is our program manager for the community part. And we have a specific focus in Hope to use actually arts to communicate the concept of block, lock, and excise. And this is done with Pauline Sameshima, who is a professor at Lakehead in California. And this is actually a picture of people making art together. And we did this in our last annual meeting, where we had everybody sitting together, community, researchers, and doing art together to better understand the complex HIV cure research concepts, and also for the researchers to better understand what it is like living with the infection. We have a lot of outreach. We have a very active community advisory board. This was an event that was sponsored by Hope during the last HIV cure meeting in Miami. The Red event was fantastic, great attendance and a wonderful program. We are very active in social media, especially Raif here, Derazi, has a YouTube channel, and he has done a lot of movies and films about block, lock, and excise. And here, of course, is your own Vivian, who has built a fantastic podcast interviewing Monica Gandhi, here part of our scientific advisory board, and a leading clinical HIV researcher on HIV cure. The Sao Paulo group is really an amazing, important part of our community outreach, incredibly active and involved, and really a joy to have with us. And we also have strong participation from our side in Kampala, Uganda. I think you're all aware of that. Uganda is going through a very tough time politically currently in terms of HIV-related politics, and so there's a lot of support that we're currently pouring into the community there in Uganda, and we hope that the policies and the laws are going to be reversed. So with this, I end. I think I thank a wonderful Hope community that we have, that is really very international, but also very multidisciplinary, from different scientific disciplines, from basic to clinical, from research to community. There's really everybody represented and bringing their best forward in order to push this new concept of block, lock, and excise forward. Also want to thank my lab. It's a really fantastic group. We're working on different viruses, but a large part of the lab is devoted to HIV cure. You see Ursula here, you see Sejong here, you see Francisco here, and Daniela was not bowling with us on that day, but she is zoomed in. If you want to learn more, we have a website. There's a great new YouTube movie explaining block, lock, and excise. And we're also very active on Twitter with this, I think. I stop and I thank you for your attention, and I'm happy to take any questions.
Thank you, Melanie. It was a great presentation, very exciting, fascinating science that we are all working together on. I want to first ask if we have questions from the audience. Esper, do you want to comment?
No, I couldn't be more excited. I think for several reasons. The first is Jessica and Melanie have so much beautiful slides. I mean, these are like amazing presentations that is a beyond lecture to all of us, how to put together this such fantastic slideshow and portray the results and discussions and guide everybody through what is a way to communicate. I think this is really amazing. Thank you for doing this. I think these two presentations bring together one aspect, which is chronic consequences of HIV infection, and they go side by side, although they look at different aspects of HIV infection and how they result in problems to the host. And to me, looking at HIV cure or a block, lock, and excise is we are very confident and very, very hopeful that this will work. But in the end, what you need is to apply this to the more elderly population who lives with HIV for such a long time. And this is going to be a challenge. And understanding the intricacies of HIV integration in those individuals that are suffering from aging and accelerated by the HIV, it's going to be really critical down the line. I hope we have that problem to solve, and but certainly we need to start thinking more carefully, or continue to think carefully about this in action.
I couldn't agree more, Esper. I think we're very fortunate that we have such a strong connection with the community here, because that also allows us to learn more about what they want and what they don't want, and what they understand and what they don't understand. And I think it's really important to bring the community along here, elderly, young people, to sort of understand what are the risks, the remaining risks that are there, especially when you're young. You might say, well, I don't care about aging because that's not my major problem currently. But I think the long-term risks are really real. And I think I found it specifically exciting to see that health span, component here, because that's what aging is about, or anti-aging is currently all about, to increase health span instead of just simply lifespan. And I think that is something that is very important to communicate, because you could argue that, okay, well, my treatment is perfect, ART is fine, and I can manage, I have no virus detectable, and I have no symptoms. But I think the key is that the research is really showing us that it's not completely fine. It's so much better, but it's not completely fine. And we have to find the right way to actually include different things into the current regimen, so that we prevent that rebound later to happen and can take people off treatment, and we don't have that residual virus activity that fueled the inflammation and the aging. So I think I couldn't agree more that this is the important pillar that we all in the science are often trying to overlook, that you need to bring the community along, because you can make a vaccine like we have seen for SARS-CoV-2 and nobody takes it. Or, you can make a drug to cure HIV and nobody will take it. And I think that is, a very, very important aspect of what we're doing. And that's where, the contact with the clinic and the contact with the community is really very, very critical for us. And we learned a lot of things. We learned that people don't want to be cured, because cure HIV status confers membership to a certain community that they get expelled of when they are cured. We have 5 cured patients, and they have all reported the same. And I think there's also a fear to lose services, because especially here in the United States, you know, HIV status is also connected to certain services that you get, medical and housing and other things. And that is very important for a certain group of people. And so I think these are all variables that we have to be very careful with, to talk about a cure, for example. When is a patient cured versus, you know, somebody who's managing without controlling, without treatment. I think these will be very, very important to define in order to bring the community on board, and without jeopardizing what they have.
That's wonderful, Melanie. I couldn't agree more. We had several discussions on that within the HOPE community group, and that's eye-opening, I think, for all of us who are working with the science and in the clinic.
Totally. Yeah, we are very grateful for this feedback.
Do we have any additional questions from the audience, Angela?
Alguma pergunta, gente? Melanie? Pode ser em português. A Vivi traduz para inglês. Eu acho que eu vou fazer alguns comentários, Vi. Se você puder traduzir para Melanie, por favor. Eu queria agradecer pela excelente apresentação. Foi fantástica. Eu acho que é um tema muito complexo, né? Que tem um conteúdo que vem muito aí da ciência básica e que se aprofunda demais na parte da ciência básica e que muitas vezes é bem complexo para a gente, até mesmo quem já tem alguns anos de trabalho com HIV na clínica, compreender completamente, mas que começar a se aproximar desse tema, né, a gente conseguir ter uma palestrante do nível da Melanie, com essa qualidade toda, e conseguir ter essa apresentação gravada para a gente poder acessar depois e tentar, por repetição, rever, conseguir compreender melhor, vai ser muito importante, vai ser realmente um grande privilégio que a gente vai viver nesse momento, né? De poder, aqui agora nos próximos meses, ter acesso a todo esse conteúdo e rever e aprender, né?
Só um minutinho, Angela, deixa eu traduzir aqui. So, Melanie, Angela says, thank you for the lecture. It was really wonderful, very clarifying. And for us in the clinic who are not familiar with bench work, the strategies may be a bit challenging to understand, but it was a privilege to have you talking with us and recording. We have this recorded presentation also for all of us in the clinic to watch over and over and try to understand all the concepts that have been shown.
Thank you. Guys, don't be shy. This is not too complicated. This is very easy. This is just like the clinic, you know. It's just a different angle, you know. But we're working around the same thing, you know. It's like a few things that you put together, and then you have to move these players around so that your result at the end is what you want. But if you understand, if you got away with, you know, this core transcriptional complex that I think is at the core of it, I think it will be easier to understand in the future. And don't hesitate to reach out if you have any questions.
E Vivi, eu gostaria de aproveitar esse momento também para pedir para você dar uma palhinha para a gente contar um pouquinho do papel que você tem desenvolvido no HOPE como podcast, né? E como que você tem enxergado essa participação, né? Que você vindo aqui junto nessa parceria entre a USP e o HOPE tem desenvolvido essa etapa aí de comunicação dos resultados e do programa de tentar e chegar na cura do HIV. Traduz só antes para Melanie, que eu pedi, por favor.
Oh, Melanie, Angela is now asking me to comment on my participation with the podcast and the animation, sharing with our colleagues the experience that we had. So I'm now going to switch to Portuguese to talk to them.
Please, I think she's doing amazing work.
Então, pessoal, esse desafio que vocês viram, né, de assistir uma aula a respeito de epigenética, certamente vai ser muito maior para pessoas leigas, pacientes, e outros profissionais de saúde que têm ainda menos intimidade com esse assunto para conseguir entender, né? E percebendo isso, quando a gente conversava com as pessoas, aí a gente explicava da estratégia de block, lock, excise, que é justamente você tentar silenciar, bloquear completamente a replicação do vírus e até removê-lo, né, do material genético. Não é um conceito simples de se entender, né? E aí, percebendo isso, a gente, durante as reuniões do HOPE, a gente também tinha participação de membros da comunidade, e aí existia também essa dificuldade de traduzir, né, de lá para cá, de cá para lá, os conceitos, né? Então, algumas estratégias de comunicação que a gente pode utilizar e que a gente consegue, com muita facilidade, divulgar amplamente, né? A gente criou uma animação mostrando como é que funciona Block, Lock, Excise, de forma bem simples. Essa animação está disponível no YouTube, posso passar o link para vocês. E o podcast, que é uma coisa que eu venho trabalhando já desde 2020, é uma estratégia de trazer para os ouvidos das pessoas informação em saúde, e nesse caso especificamente informação em relação ao HIV, as estratégias que a gente já desenvolveu nos últimos 40 anos, para tratamento, prevenção e agora cura. É uma iniciativa com a qual eu venho trabalhando. A gente produziu uma temporada inteira com 6 episódios, 5 em português, mais 1 em inglês, falando sobre HIV. E eu tenho muito orgulho de ter participado disso. É um trabalho que ajuda muito mais pessoas a conseguir entender essa ciência, o quanto ela é fascinante. E o que eu tenho mais esperança é que isso ajude também a diminuir o preconceito e o estigma das pessoas, para as pessoas que vivem com HIV.
Vivian, please, we need a right of voice and make the two presentation.
Could you repeat, Dr. Valdir? I couldn't understand.
A gente precisa do direito de sua imagem das duas apresentações.
Ah, tá bom. Melanie, we're going to ask for your signature on the authorization to use the recorded lecture for our internal team to be able to access. Is that okay for you?
Yes, I mean, there's several unpublished stuff in there, but I think if it's only internal, I think it's fine.
Thank you.
Alguma pergunta, algum comentário, pessoal? Não?
Melanie, thank you so much once again.
My pleasure. Good to meet you all, and keep up the good work. Bye.
Bye, everyone. Thanks again.
Bye. Thank you.